In data science, a larger sample size inevitably sharpens the resolution of insights. However, high-volume data influxes can sometimes cause the exact opposite effect. The deployment of the COVID-19 vaccines was the largest and fastest population-scale pharmaceutical rollout in human history. Regulators monitored this rollout using the Vaccine Adverse Event Reporting System, or VAERS. For 3 decades, this passive early warning system operated at a predictable and relatively low historical baseline. In 2021, the system logged 18 times its historical maximum reporting number in a single year. Standard biostatistical logic suggests that a larger N inherently improves signal-to-noise ratios. 1 million data points should, in theory, provide a high-resolution map of emerging risks. Instead, the 18-fold surge in reports compromised the ability of biostatisticians to isolate specific drug risks during the rollout. The structural breakdown of proportional reporting ratios and early warning architectures suggests that extreme data volume requires a corresponding evolution in detection logic. When passive surveillance systems face extreme volume surges, they require a specific type of algorithmic adaptability to remain functional. We need to examine the mathematical model of how these datasets interact to understand why the existing system stalled. Detecting safety signals against a massive background database relies on a statistical method called empirical Bayesian data mining. This method follows a baseline comparison rule. The algorithm flags a statistical signal only if a drug's adverse event rate significantly exceeds the historical background rate. This graph illustrates standard signal detection. If we compare a toxic substance against a non-toxic baseline, like T, the drug's effects appear as a sharp data spike rising high above the detection threshold. If we then integrate a second, similarly toxic dataset into that background control group, the baseline inflates. As the baseline rises, the relative distance of the signal shrinks until it falls below the algorithmic detection threshold. The signal is still there, but the math can no longer see it. This exact model explains the real-world deployment of multiple mRNA platform vaccines. Because these vaccines used the same platform and shared similar adverse event profiles, their concurrent rollout flooded the passive surveillance database with nearly identical data. When the algorithm looked for myocarditis signals in vaccine A, it compared those reports against a baseline heavily populated by vaccine B's own myocarditis cases. The sheer volume of this homologous data washed out the proportional reporting ratio analytics used by the CDC and FDA. This is statistical masking. It occurs when a significant safety signal is erased because the background noise is need of the exact same signal. While passive surveillance faced algorithmic masking, active surveillance systems, which solicit data directly from participants, encountered a different structural failure. Regulators launched vSafe to collect high-fidelity health data from millions of active volunteers. The application's user interface restricted structured data inputs to minor, inconsequential symptoms like fatigue, headache, and chills. Severe or complex endpoints were not programmed into these easy-to-track checkboxes. Participants experiencing more serious conditions were forced to bypass the structured checkboxes and use unstructured free text fields to describe their health status. Roughly 1/3 of the 10 million volunteers used these free text fields. This created a massive data bottleneck, as unstructured text requires manual review or intensive, delayed processing compared to instantaneous checkbox data. Hidden in this backlog were hundreds of manual entries for myocarditis and tens of thousands of reports of chest pain. Because this data was unstructured, it could not be queried or analyzed with the same velocity as the minor symptoms. Passive systems were hindered by masking, while active systems were slowed by a UI design that delayed warnings until after hundreds of millions of doses were administered. Restricting inputs in an active surveillance system forces critical biological data into computationally opaque formats, undermining the mandate for an early warning. Adaptive software protocols exist specifically to address the problem of statistical masking. A demasking algorithm acts as a filter. It identifies and excludes confounding, homologous drugs from the baseline denominator during a data run. Once the baseline is restored to its proper level, the previously obscured safety signal reemerges above the detection threshold. Veteran statisticians communicated these specific algorithmic fixes to the FDA using established commercially available software. Regulators declined to implement the fix, citing concerns about using newly modified or unvalidated tools during an emergency use authorization. Bureaucratic adherence to standard operating procedures continued, even as the volume of homologous data rendered those procedures mathematically ineffective. The institutional friction of updating analytical models during a crisis prevented the detection of safety signals, despite the availability of both the necessary data and the software to process it. Regulatory institutions are responsible for maintaining the integrity of the data architectures used to monitor novel drug deployments. We now know that static comparative baselines cannot accurately process simultaneous rollouts of similar drug platforms. We also know that UI architectures that exclude anticipated severe endpoints create data bottlenecks that delay critical public health responses. A modernized early warning system requires a different blueprint. Data from both active and passive sources must flow through a dynamic demasking filter to ensure relative comparisons remain accurate. This architecture requires flexible active surveillance endpoints that can adapt to include severe adverse events as they are identified. Automated demasking must be integrated as a standard procedure to prevent massive influxes of homologous data from washing out the signals we are looking for. Pharmacovigilance networks require dynamic computational adaptability. Ensuring that these systems can process unprecedented data volumes is essential for maintaining the integrity of public health monitoring.